Imagine this. A 5-year-old male immigrant who was immunized with Bacillus calmette-guerin, BCG, at birth, undergoes a tuberculin test at a physician's office. The results show a less than 4 millimeter wide indurative spot. What do you think is the underlying mechanism of this false positive test? Could it be IgG binding cell surface protein? This mechanism involves the binding of IgG antibodies to cell surface proteins, triggering an immune response. However, this is not the reason for our false positive result. Perhaps it is IgE binding antigen. This mechanism involves IgE antibodies binding to specific antigens, leading to an allergic reaction. Again, this is not the case here. The tuberculin test does not measure IgE antibodies or allergic reactions. Could it be complement fixation? This mechanism involves the activation of the complement system, which aids antibodies and phagocytic cells to clear pathogens from an organism. However, this is not the reason for the false positive result. What about T-cell mediated? This mechanism involves T cells, a type of white blood cell that respond to specific antigens. In the case of the tuberculin test, it measures the cell-mediated response to the tuberculin antigen, which is why it is used for diagnosing tuberculosis. However, the BCG vaccine, which the child received at birth, can also trigger a positive reaction because it contains a live, weakened strain of Mycobacterium bovis, which is similar to Mycobacterium tuberculosis. Therefore, the T-cell mediated response is the underlying mechanism of the false positive result. Lastly, why is it not due to a specific antigen-antibody ratio? This mechanism involves the balance between the amount of antigen and the amount of antibodies. A false positive result could occur if there is an imbalance. However, this is not the case here. The tuberculin test measures a cell-mediated response and not an antibody response. In summary, the false positive result of the tuberculin test in a child who was immunized with BCG at birth is due to a T-cell mediated response. This response is triggered by the similarity between the antigens in the BCG vaccine and the tuberculin antigen. The other options, IgG binding cell surface protein, IgE binding antigen, complement fixation, and specific antigen antibody ratio, are not the underlying mechanisms because they do not involve a cell-mediated response, which the tuberculin test measures.